Greetings, everyone. I'm Tom O'Halloran. I'm the director of the Northwestern University PSOC, and I just want to join everyone in congratulating NCI and the staff, the Board of Scientific Advisors, for over 12 years ago taking a risk in this high risk, high reward space of bridging two disparate scientific areas across the physical sciences and oncology. This is just a great effort led by John Niederhuber and Anna Barker in the beginning. Naz Zahir and Sean Hanlon are some of the people that helped us put this together, and it's just been an extraordinary run. I'm going to quickly tell you about the Northwestern Science Oncology Center. John Whittam and I started it really based on his discovery that there's a fourth level of the genetic code, that the local sequence triplets and non-coding regions had a profound effect on binding of nucleosomes to some sequences and not others. And this then, as we've all learned over the years, controls the local topology and the excess and patterns of gene expression. And then in cancer, these things are disrupted. And not only does disruption of this information flow transfer, can it be read in the sequence data and chemical signaling, but it also has a huge impact on nuclear structure, nuclear organization, which then is influenced by chemotherapeutics as we try to convince and move these cancer cells back to a normal state. And our cancer center at Northwestern, Comprehensive Cancer Center, and the Chemistry of Life Process Institute collaborated to bring teams together. We've had just an incredible run over the last 10 years, over 40 investigators, 130 graduate students and postdocs trained, many of whom have gone on to faculty leadership positions in oncology and the physical sciences, over 200 great papers, numerous undergraduates moving in, and all this across an array of institutions. It could never have happened without really NCI's persistent support of this joint venture. I mentioned John's discoveries and his work, then subsequent work with Eran Segal, Rob Phillips, then bringing, after John's unfortunate death in 2011, really the team that he and I organized then began attacking and trying to understand how these levels of information control impacted cancer, really how to go in and intervene in new types of chemotherapy. And with my colleague Licht at the University of Florida Health Cancer Center, really help introduce new concepts into both for the oncologists as well as the physical scientists. And one of our legacies is just an entire array of new types of microscopy that go down to 6 nanometers and millisecond time scale and live cell imaging without labels. So these are some of the legacy aspects of our program. Some of the most important things that we've done is train the next generation of people who are going to go out and apply these new ideas and methods to really improve patient care. And so my colleague, Dr. Sheila Judge, has really left us a variety of really incredible training resources. I'm going to stop here, introduce John Licht, a clinician and director of the Cancer Center at University of Florida, basically to tell you how he's applied some of these ideas in his work. I'll stop there. Thank you.
I would like to present the results of a PSOC collaboration over many years taking place both in Chicago and in Florida. The uncovering of histone mutations that affect the histone folds. As seen in this diagram here, a mutation that causes a clash between the histone H2B and H4 proteins, destabilizing the nucleosome. When this muted histone is expressed in mammalian cells, chromatin opens up, as seen by this increased susceptibility to micrococcal nuclease digestion. Using more modern attack-seek methods, thousands of new promoters are opened up and accessible for transcription in the presence of the mutant histone. Histone mutations affecting the folds of histones and chromatin integrity may be found in between 5 and 10% of cancers of a variety of types, particularly those associated with smoking, such as cervical, bladder, lung, and head and neck cancer. These mutations are subclonal. They occur after the initial cancer drivers have occurred in tumors and are commonly co-occurring with such cancer drivers such as RAS, P53, and PIK3CA. In fact, in the laboratory, co-expression of mutant H2B in collaboration with mutant PIK3CA causes enhanced formation of tumorigenic colonies in soft agar assays. What are the clinical implications of these findings? We believe that histone mutations that compromise the physical integrity of chromatin may alter gene expression and expand oncogenic pathways expressed in tumors. We have preliminary evidence that these mutations diversify gene expression and could contribute to increased tumor heterogeneity. And we believe destabilization of the genome could alter mutation rates, rates of DNA repair, and contribute to the development of tumor resistance to therapy.